13 million.
Yeah.
That is the number of people who will die this year, and not from an inherited genetic flaw, right?
Mm.
Not from old age, but simply because of the physical space they inhabit. It's a mortality statistic. It fundamentally reframes how we should view pathology.
It really does, because the World Health Organization data we're examining today makes it incredibly clear that the environment is not just some passive backdrop to human life. It's an active and honestly often aggressive participant.
Right.
Those 13 million annual deaths, they're attributed entirely to preventable environmental risks. We're looking at unsafe drinking water, a lack of basic sanitation, and the pervasive hazard of indoor air pollution from, the combustion of solid cooking fuels.
Which is staggering.
And crucially, 4 million of those casualties are children.
Wow. 4 million.
Yeah.
As clinicians or medical students, we're basically trained to treat the fish.
Treat the fish, yeah.
Right, like a patient presents with a pathology, we isolate the physiological dysfunction, we prescribe the pharmacological intervention, or we perform the surgery. We focus entirely on the organism right in front of us.
Exactly, the immediate host.
But environmental health it's upstream engineering. We can't just keep treating the fish, We have to test the water. We need to analyze the environment itself as this sort of invisible scaffolding of human health.
That's a great way to frame it.
So for everyone listening right now, whether you're prepping for your boards, completing a public health fellowship, or just managing patients in a busy clinic, this deep dive is designed as your companion to Chapter 7.1 of the Oxford Textbook of Global Public Health.
And our mission here isn't to simply recite facts you could just read yourself.
Right. No one wants that.
Exactly. We are building clear operational frameworks. We're going to trace the logic from the initial hazard to the exposure down to the molecular outcome and finally to the systemic intervention.
So what's our roadmap look like?
Well, the source material provides a really rigorous blueprint for that progression. We'll trace the historical evolution of how society even defines a cause of disease, which shifted dramatically as we industrialized.
Right.
We'll unpack the structural models of host-environment interaction, move through the hierarchy of epidemiological study designs from massive aggregate populations all the way down to specific molecular biomarkers, and ultimately examine how public health infrastructure can literally engineer disease out of existence.
Okay, to build that framework, I feel like we have to look backward first. Because the way a 21st century epidemiologist views a disease cluster is, it's light years away from how early physicians approached the same problem.
Absolutely.
Yet the foundational instinct that the environment dictates health goes all the way back to antiquity, right? Like we see it in Hippocrates.
We do. In his classical text, On Airs, Waters, and Places, Hippocrates was actually already documenting the profound influence of seasons, prevailing winds, the geographic orientation of a city, and even the mineral quality of the water supply on endemic disease rates.
Right, he was noticing patterns.
Exactly. He noted that certain pathologies clustered in specific topographies and at very specific times of the year.
Which is amazing because he obviously didn't have a microscope to see a pathogen.
No, not at all.
But he had the clinical observation skills to recognize geographic and temporal clustering.
Yeah.
The challenge was that while the observation was totally correct, the biological mechanism was completely missing.
Right. There was a massive gap in understanding.
And that vacuum of mechanistic understanding gave rise to early explanatory models. Right?
Yeah.
Primarily the contagion theory and the miasma theory.
Yeah, so the contagion theory operated on a fairly straightforward premise, which is that disease is transmitted through direct or indirect contact with sick individuals or animals.
Like catching a cold.
Right. And the public health interventions born from this were isolation and quarantine. We saw this extensively during the plague years with the quarantine of ships. But contagion theory failed to explain why massive outbreaks would suddenly erupt in populations with no obvious external contact.
Oh.
Or why certain diseases seem to just rise from the slums themselves with no obvious patient zero.
Which paved the way for the miasma theory to take hold. This is the idea that decaying organic matter, dirty living environments, and poor ventilation basically created miasma, literally bad or polluted air, that directly cause disease.
Bad air, exactly.
I always find the miasma era fascinating because, clinically speaking, the biological mechanism was wildly incorrect for diseases like cholera. Bad smells do not cause bacterial infections.
No, they don't.
But the interventions the theory inspired were actually revolutionary.
It's one of the great paradoxes of public health history, really. Miasma theory drove the great sanitary awakening of the 19th century.
Right.
In the UK, Edwin Chadwick's crusade against filth led to the landmark Public Health Act of 1848. This was essentially the birth of municipal hygiene.
Because they were trying to get rid of the smell.
Exactly. Governments began constructing these massive sewage disposal systems, mandating better ventilation in housing, securing safer drinking water supplies. And by doing that, they drastically reduced the incidence of waterborne and vector-borne diseases.
Wow.
They engineered the environment perfectly, even though their underlying theory of pathogenesis was completely flawed.
It just proves that in public health, an effective intervention doesn't always require a perfect molecular understanding of the agent.
That's a really key point.
But that understanding did eventually become necessary. And the pivotal transition point is always traced back to London, 1854, and John Snow's investigation of the Broad Street cholera outbreak.
The famous pump handle.
Right. But here's the critical nuance that often gets glossed over, I think. Removing the pump handle didn't prove the existence of a germ.
No, it didn't. It proved the vehicle of transmission.
Right.
Snow's epidemiological map was an exercise in environmental hazard identification. He plotted the geographic distribution of cholera deaths and found this really dense cluster around the Broad Street water pump.
And then he actually went and talked to people, right?
Yeah. He conducted a rudimentary case control study. He interviewed the families of the deceased and confirmed that they drank from that specific pump. So by removing the handle, he halted the environmental exposure. And he accomplished this decades before Robert Koch conclusively identified the Vibrio cholerae bacterium.
So Snow's work sort of bridged the gap between the environmental interventions of the miasma era and the microbiological precision of the germ theory era.
Perfectly stated.
Because by the time Robert Koch published his famous postulates in 1891, the paradigm had entirely shifted. We were suddenly looking for the singular microscopic assassin. Koch demanded a strict one-to-one necessary and sufficient relationship.
Right. One specific germ causes one specific disease.
Okay, let's unpack this for a second.
Right.
Koch's postulates require that the microorganism must be found in abundance in all organisms suffering from the disease, but shouldn't be found in healthy organisms.
Correct.
It must be isolated and grown in pure culture. The cultured microorganism should cause disease when introduced into a healthy organism, and then it must be re-isolated from the inoculated diseased experimental host.
It's very rigorous. And for acute infectious diseases like tuberculosis or anthrax, this framework is an absolute masterpiece of biological proof.
Sure. But as a clinician today, if a patient walks into my clinic with ischemic heart disease or, say, a neurodegenerative disorder, Koch's postulates are entirely useless to me.
Completely useless.
Right. I cannot culture the singular heart disease microbe in a petri dish.
Yeah.
And this is basically the result of the epidemiological transition.
Yeah, the 20th century saw the widespread deployment of the very interventions Koch and his contemporaries pioneered, right?
Mm-hmm.
Vaccines, targeted antibiotics, vector control.
Like spraying for mosquitoes.
Exactly. Our sources actually highlight the use of the insecticide DDT to completely eradicate malaria in Taiwan as a prime example. And as infectious diseases were brought under control and infant mortality plummeted, life expectancy increased dramatically.
Which is great, obviously.
It is. But the demographic and epidemiological profiles of populations aged, and the burden of disease shifted heavily to chronic, non-communicable diseases.
We basically traded acute microbial threats for insidious environmental and lifestyle threats.
Yes.
We're dealing with cardiovascular diseases, metabolic syndromes, malignancies, and the pathology of these diseases is fundamentally multifactorial.
Extremely multifactorial.
Take an environmental hazard like arsenic in drinking water. It doesn't just cause one thing. The literature shows it induces skin, bladder, kidney, and lung cancers, alongside hypertension, diabetes, and cerebral infarction.
It's highly pleiotropic. One environmental agent drives a massive spectrum of systemic dysfunction.
And the inverse is also true, isn't it?
Okay.
A single clinical outcome, like ischemic heart disease, can be driven by a multitude of different environmental and host factors.
Oh, definitely. Air pollution, heavy metal exposure, psychosocial stress, hyperlipidemia, smoking. Many different agents can converge on one single disease.
So the one-to-one rigidity of Koch's postulates simply breaks down in the face of this complexity.
It completely collapses.
It's like we went from searching for a single smoking gun at a simple robbery scene to prosecuting a massive, decades-long corporate conspiracy.
I love that analogy.
To prove an environmental exposure causes a chronic disease, We needed a completely different evidentiary standard. We needed Sir Austin Bradford Hill.
Yes. Hill's criteria for causation, which he formulated in 1965, recognized that in chronic disease epidemiology, we just cannot rely on a single laboratory experiment. We have to weigh a totality of circumstantial and observational evidence.
So what are his key viewpoints?
Well, he outlined several. There's the strength of association, meaning how massive is the relative risk between the exposed and unexposed. Then there's consistency. Has this association been repeatedly observed by different persons in different places, circumstances, and times?
Specificity is in there too, though as we just discussed with arsenic, environmental toxins are rarely specific to one disease.
Right. Specificity is often the weakest criterion for environmental health.
But temporality is absolute.
Absolutely. The exposure must precede the disease.
And perhaps the most compelling for environmental exposures is the biological gradient, or the dose-response curve. Like, if a patient drinks a little contaminated water, their risk is mildly elevated. If they drink it every day for 30 years, their risk skyrockets.
Exactly. Hill also emphasized biological plausibility and coherence. The epidemiological association shouldn't violently conflict with the generally known facts of the natural history and biology of the disease.
Right, it has to make biological sense.
Exactly. If we see a statistical link between a heavy metal and neurological decline in a population, we have to ask, does that metal actually cross the blood-brain barrier in animal models? Does it disrupt neurotransmitter synthesis in vitro? The observational data and the laboratory biology really must align.
Understanding this multifactorial framework is paramount because modern environmental health hazards operate on long, quiet timelines.
Very quiet.
Now, yes, we still have acute catastrophic failures. The text cites the 1984 methyl isocyanate gas leak in Bhopal, India, and the 1986 Chernobyl nuclear disaster. Those events produced horrific acute casualties. But the public health burden of those disasters extends decades into the future, with chronic respiratory morbidities and thyroid carcinomas.
Right, and furthermore, the vast majority of environmental pathology is driven by slow, continuous exposure. Industrial waste provides the most tragic examples of this.
Like what happened in Japan.
Yes. In Japan, we saw itai disease, which is a severe osteomalacia and renal tubular dysfunction. That was caused by chronic cadmium poisoning from mining runoff that was polluting the Jinzu River basin.
Wow.
And we also saw Minamata disease, a devastating neurological syndrome caused by severe methylmercury poisoning from industrial wastewater. That mercury bioaccumulated in the local fish and shellfish populations.
So the food supply itself was toxic.
Exactly. These populations were poisoned meal by meal, sip by sip, over years.
So the environment acts as a vector for these physical, chemical, and biological agents. And to manage this, public health professionals need to conceptually map how these agents interact with the human host.
They need structural models.
Right. If we're abandoning Koch's simple one-to-one model, what structural models do we use to visualize disease pathogenesis?
The sources outline 3 distinct causal models, and each serves a different conceptual purpose. The first, and probably the most traditional, is the epidemiological triangle.
Okay.
The vertices of the triangle are host, agent, and environment. The model basically posits that disease is the product of the interaction among all 3. It's an excellent framework for identifying intervention points.
Can you give an example?
Sure. If there's a malaria outbreak, you can target the host with prophylactic medication. You can target the agent by developing therapies against the plasmodium itself, or you can manipulate the environment by draining stagnant water to reduce the mosquito vector.
So the triangle is highly pragmatic for infectious disease control. But structurally, it still relies on the premise of a unique, necessary agent.
Yes, it does.
Like without the plasmodium, there is no malaria. But for something like hypertension, there is no single necessary vertex you can just label agent.
Which necessitates the second model, the ecological wheel.
Right.
The wheel conceptualizes the host's genetic core as the central hub. Surrounding that hub are the host's innate characteristics, their immune baseline, their psychological profile. And forming the outer tire of the wheel is the environment, explicitly divided into the physicochemical, biological, and social environments.
The genius of the ecological wheel is what it deliberately omits. There is no isolated agent node.
Exactly.
It forces the clinician or researcher to look at the holistic balance of exposures. Think of it like this. If a patient has a massive genetic predisposition to a condition, the genetic core expands, and it requires very little environmental push to trigger the disease.
Right.
Conversely, In the case of severe environmental trauma or overwhelming toxin exposure, the environmental sectors dominate the wheel, overriding even a really robust genetic core.
That's spot on. The limitation of both the triangle and the wheel, however, is that they are static. They really only represent a single moment of pathogenesis.
Right, like a snapshot of a cell.
Exactly. But chronic disease is a longitudinal process. A carcinogen doesn't mutate a cell and produce a palpable tumor in the same afternoon.
It takes years.
Decades, often. Therefore, the most sophisticated model for chronic environmental health issues is the evolutionary spiral.
The evolutionary spiral takes the ecological wheel and essentially extrudes it through the dimension of time. It's like a time-lapse video.
I like that.
It maps the multi-stage sequence of pathogenesis, initiation, promotion, progression, invasion, and clinical manifestation. And the textbook uses the development of hepatocellular carcinoma liver cancer as the archetypal example of this spiral.
Hepatocarcinogenesis beautifully illustrates the temporal complexity. We begin at the origin of the spiral, a healthy host. The initial event is an infection by a biological agent, the hepatitis B virus, or HBV.
Right.
For a period, the host may simply be an asymptomatic carrier. But as time moves forward, spiraling outward, the virus initiates chronic hepatitis. This constant inflammatory state leads to liver fibrosis, which advances to cirrhosis, and finally to hepatocellular carcinoma.
But the virus isn't operating in a vacuum for 20 years, right?
Not at all.
As that spiral expands, the host is constantly interacting with their physicochemical and social environments. Their liver is processing dietary chemical carcinogens, like aflatoxins or benzoaperine.
Yes.
Their social environment dictates whether they have access to an abdominal ultrasound screening or antiviral therapies that could interrupt the spiral. Their genetic core dictates how efficiently their cellular immunity controls the viral replication. The evolutionary spiral accommodates all of these variables interacting at different stages over decades.
And understanding that a chronic disease takes decades to spiral into existence fundamentally changes how we must conduct surveillance.
How so?
Because a single clinician seeing a single patient cannot easily perceive an environmental hazard that has been acting for 30 years. To identify these hazards, public health must deploy macro-level surveillance. We have to look at entire populations simultaneously.
Which brings us to the methodology of ecological studies. When we talk about ecological studies in epidemiology, we are talking about aggregate data.
Right.
We are no longer looking at individual patient charts. We are looking at townships, counties, or entire nations. We are asking, does a specific geographic area have an abnormally high rate of a specific disease? And does that same area have a high prevalence of a specific environmental exposure?
The unit of observation is the group, not the individual. And the most profoundly illustrative example of this in the environmental health literature is the investigation of Blackfoot disease on the southwest coast of Taiwan.
The clinical presentation of Blackfoot disease is harrowing.
It's awful.
It's a severe peripheral vascular disorder. Patients initially present with numbness, coldness in the extremities, and intermittent claudication, which is severe cramping pain in the legs when walking due to restricted blood flow.
Right.
As the ischemia progresses, tissue necrosis sets in. The skin turns black, severe ulcers form, and gangrene develops. In the terminal stages, the affected fingers or toes undergo spontaneous amputation.
It was an agonizing, debilitating condition, and epidemiological surveillance revealed it was hyperendemic. But it wasn't spread across the entire country. It was strictly confined to a cluster of 4 neighboring townships on the coast.
And that intense geographic clustering is the ultimate red flag for an environmental point source.
Exactly. The researchers began investigating the history of the built environment in those 4 townships. They discovered a massive infrastructural shift that occurred in the 1920s.
Okay.
The shallow wells the residents had historically used for drinking water were highly saline and unpalatable. So the communities began digging deep artesian wells, penetrating 100 to 200 meters into the earth to access fresh water.
They just wanted better tasting water.
Right. But the researchers hypothesized that this deep well water was the vehicle for the causal agent.
So to test this hypothesis at the aggregate level, they utilized the type of well water as a surrogate indicator for the exposure. They analyzed the water and found staggering concentrations of arsenic ranging from 0.35 to 1.14 milligrams per liter.
Extremely high levels.
But they didn't just look at Blackfoot disease. They calculated standardized mortality ratios, or SMRs, for the entire population of those townships. An SMR essentially compares the mortality rate of a specific study population to the mortality rate of the general population, adjusting for age and sex.
And the SMRs revealed a secondary hidden epidemic. The residents of the Blackfoot endemic area had mortality rates for cancers of the bladder, kidney, skin, and lung that were roughly 3 times higher than the general population of Taiwan.
3 times higher, that's huge.
It's a massive signal. The ecological study then sought to establish a biological gradient at the aggregate level. Because the prevalence of Blackfoot disease varied among the 4 townships, researchers plotted the township-specific prevalence of Blackfoot disease against the township-specific standardized mortality ratios for those cancers.
And what did they find?
The correlation was nearly perfect. The higher the rate of Blackfoot disease in a township, the higher their mortality from bladder, kidney, and lung cancers.
And they zoomed in even further, right? Looking at 84 specific villages within the endemic area.
Yes.
Villages that relied exclusively on the deep artesian wells had the highest cancer mortality rates. Villages that used a combination of artesian and shallow wells had intermediate mortality rates. And villages that only used the shallow wells had the lowest mortality rates.
It was a massive, naturally occurring dose-response curve mapped across an entire region.
And later, an expanded ecological study looked at over 300 precincts and townships across all of Taiwan. They tested more than 83,000 wells to calculate the median arsenic concentration for each township.
Just a huge undertaking.
Right. And cross-referencing this massive environmental data set with national death registries, they found a highly significant positive correlation between a township's average arsenic level and their mortality rates for a wide spectrum of malignancies, including liver and prostate cancers.
The scale of that aggregate data is just breathtaking. It acts as an incredibly sensitive early warning radar system. It scanned the topography of the nation and pinpointed the exact location of the hazard.
But as an epidemiologist, looking at group averages introduces a massive conceptual vulnerability. We have to address the ecological fallacy.
Yes, the ecological fallacy is the critical limitation of aggregate data. An ecological study identifies a hazard within a population, but it cannot definitively prove the risk to an individual within that population.
Right.
The fallacy occurs when a researcher assumes that the statistical correlation observed at the group level perfectly and uniformly applies to every individual member of that group.
So what does this all mean for a researcher? If ecological studies carry the risk of the ecological fallacy, why do we even use them? Let's ground this in a clinical scenario. The aggregate data shows that Village A has a high median arsenic level in its water and a high standardized mortality ratio for lung cancer. The ecological fallacy would be a doctor looking at Mr. Smith, a resident of Village A who just developed lung cancer, and definitively concluding the arsenic water caused your cancer. What is the missing piece of logic there?
The missing piece is individual-level confounding data. In an ecological study, you do not have data on Mr. Smith's personal habits. Mr. Smith might have been a 2-pack-a-day smoker his entire adult life.
Ah, right.
Furthermore, you don't actually know how much water Mr. Smith drank from the village well. He might have worked outside the village and consumed most of his water elsewhere. The aggregate data cannot account for individual variation in exposure, genetic susceptibility, occupational hazards, or lifestyle choices like smoking and diet.
So the ecological radar ping tells us where the danger is, but it doesn't give us the resolution to definitively convict the agent.
Exactly. They direct where individual-level studies should be deployed.
To achieve that level of proof, to control for the confounding variables of the individual, we have to deploy different study designs. We have to move from the aggregate to the individual.
And the transition to individual-level assessment involves 3 primary epidemiological designs: cross-sectional surveys, retrospective case control studies, and prospective cohort studies.
Let's differentiate those.
Sure. A cross-sectional survey collects data on both the environmental exposure and the disease status from individuals at the exact same point in time. It provides a highly accurate snapshot of disease prevalence within the population.
But the weakness of a cross-sectional study is temporal ambiguity. Because you are assessing the exposure and the outcome simultaneously, you cannot unequivocally satisfy Hill's criterion of temporality.
Right. You don't know for certain which came first.
To solve that, we use longitudinal designs that incorporate the dimension of time.
Yes. A retrospective case control study looks backward. You identify individuals who already have the disease, the cases, and you carefully match them with healthy individuals. The control is based on age, sex, and other demographic factors. Then you investigate their past environmental exposures.
Because you know they already have the disease,
Yeah.
and you're looking at historical exposure, temporality is established.
Correct. But the primary vulnerability here is recall bias. When you ask a patient to detail their dietary or water consumption habits from 20 years ago, their memory is inherently fallible. And sick patients often recall past exposures differently than healthy patients.
So then what's the gold standard?
The gold standard for observational epidemiology is the prospective cohort study. You assemble a massive group of healthy individuals, meticulously document their current baseline environmental exposures, and then you follow them forward in time for years or decades to record who develops the disease.
This yields true disease incidence rates.
Exactly. It eliminates recall bias because you are documenting the exposure in real time. The logistical nightmare, however, is the risk of participants being lost to follow up over those decades, which requires immense resources and sophisticated registry linkage to maintain the cohort.
In the investigation of Blackfoot disease, researchers systematically deployed these individual-level designs to validate the aggregate findings. They conducted a massive, community-based cross-sectional survey of over 40,000 residents, which confirmed a significant dose-response relationship between the arsenic concentration in individuals' specific household drinking water and their prevalence of both skin cancer and Blackfoot disease.
But the truly staggering validation came from the retrospective case control study. They matched 353 Blackfoot disease patients with 353 healthy controls. And what they found regarding the primary drinking water source essentially closed the case.
What were the numbers?
Every single one of the Blackfoot disease patients, 100% of the cases, had consumed artesian well water as their principal drinking source during the 15-year period prior to the clinical onset of their disease.
Wow, 100%.
Yes. When they looked at the matched healthy controls, only 66% had consumed artesian well water.
That's a massive difference.
And through detailed individual questionnaires, they were able to adjust for the confounding variables we discussed earlier: age, sex, physical undernourishment, familial disease history. Even after adjustment, the duration of artesian well water consumption remained the dominant, statistically significant predictor of the disease.
Here's where it gets really interesting, though. Because establishing that someone drank from the well is one thing, but to truly satisfy Hill's criterion of a dialogical gradient, you need to know exactly how much toxin they ingested over a lifetime.
Right.
How does an epidemiologist reconstruct a 50-year water diary for thousands of individuals to calculate risk? It seems impossible.
It's difficult, but they developed an incredibly robust metric known as cumulative arsenic exposure. In these rural villages, it was common for multiple households to share a single specific well. The researchers physically tested the water from these specific shared wells to determine a median arsenic concentration.
Okay.
Then, acknowledging that people migrate, they conducted exhaustive, structured interviews to build a complete residential history for every participant. They documented every village the person had lived in and exactly how many years they resided there.
By cross-referencing national registries.
Exactly, to overcome that individual recall bias.
So it's basically an equation of concentration multiplied by duration.
Yes.
For each period of a patient's life, the researchers took the median arsenic concentration of the well they used and multiplied it by the number of years they drank from it. They then summed all of those periods together to produce a single comprehensive number, the patient's cumulative arsenic exposure.
And armed with this precise individual-level metric, the subsequent cohort studies delivered undeniable results. The higher a patient's cumulative arsenic exposure, the higher their incidence of a vast array of chronic diseases.
The dose response was clear.
Very clear. They mapped perfect dose-response curves, linking the cumulative exposure not just to cancers and Blackfoot disease, but to diabetes mellitus, hypertension, and ischemic heart disease. They even used duplex ultrasonography to measure the exact thickness of the carotid artery intima media, proving that higher arsenic exposure directly correlated with severe carotid atherosclerosis.
So the observational data was airtight. They had the ecological radar ping, they had the individual cross-sectional prevalence, they had the case control odds ratios, and they had the prospective cohort incidence data showing a massive biological gradient.
The whole package.
But in the strict hierarchy of scientific proof, observing an association is still just observation. To definitively prove that agent X causes disease Y, you must demonstrate reversibility. If you engineer the hazard out of the environment, does the pathology disappear?
Reversibility is the ultimate test of causality in public health. But we face a fundamental ethical barrier here.
We can't intentionally poison people.
Exactly. We cannot conduct a randomized controlled trial where we intentionally administer a known toxin like arsenic to a healthy group of humans just to watch them develop cancer.
That would be horrific.
Right. Therefore, we rely on community intervention trials. These are natural experiments where the government or a public health body actively removes a suspected environmental hazard, and epidemiologists monitor the population to see if the disease rates recede.
Because it's unethical to intentionally expose people to toxins to prove a theory, these community intervention trials act as humanity's natural undo button.
That's exactly what they are. Once the artesian wells were definitively implicated, the government in Taiwan initiated a massive infrastructural intervention. Beginning in the early 1960s and concluding in the 1970s, they systematically replaced the contaminated artesian well water supply in the endemic areas.
What do they replace it with?
They built a public water infrastructure that piped in pristine surface water from a distant, uncontaminated reservoir. They fundamentally altered the physicochemical environment.
And to assess the efficacy of this massive intervention, researchers utilized cumulative sum techniques to track the cause-specific standardized mortality ratios in the decades following the water replacement.
And the secular trends were profound.
Yeah.
Over the subsequent 20 to 30 years, they documented statistically significant declines in mortality from ischemic heart disease, peripheral vascular disease, and renal disease. The mortality rates for cancers of the lung, kidney, and bladder steadily decreased in both men and women. The reversibility criterion was unequivocally met.
The hazard was removed, and the evolutionary spirals of those chronic diseases were interrupted.
Yes.
But evaluating the success of an intervention using mortality data, basically death certificates, presents significant analytical challenges for an epidemiologist, right?
It certainly does.
As a clinician, I know firsthand that what gets written on a death certificate doesn't always capture the complex web of underlying pathogenesis. Cardiopulmonary arrest is a terminal event. It's not an underlying cause.
Relying solely on mortality data introduces several critical caveats. First, the International Classification of Diseases, the ICD codes, are updated periodically. A disease might be coded differently in 1990 than it was in 1960, creating artificial fluctuations in the data.
That makes sense.
Second, because arsenic is a pleiotropic agent that causes multiple lethal diseases, we face the issue of competing causes of death. If an intervention successfully reduces the risk of dying from a rapid onset cardiovascular event, that patient might survive long enough to eventually die from a slower developing arsenic-induced lung cancer. The total mortality might not shift as dramatically as the specific disease mortalities shift.
And crucially, mortality is a function of both the incidence of the disease and the fatality rate of the disease. A drop in mortality might simply mean that oncologists got much better at treating bladder cancer with new chemotherapies rather than the environmental intervention preventing the cancer from occurring in the first place.
Exactly.
That is why tracking the decline in disease incidence, the rate of new cases, is always the most rigorous metric of prevention.
Precisely. Which brings us to another monumental intervention trial detailed in the sources, one that definitively tracked incidence. We shift from a chemical agent, arsenic, back to a biological agent, the hepatitis B virus.
Right, the HBV.
As we established with the evolutionary spiral, chronic HBV infection is the primary initiating event for hepatocellular carcinoma.
In 1984, Taiwan launched what was truly a watershed moment in global public health. The very first universal hepatitis B vaccination program for newborns in the world.
It was groundbreaking.
They initially targeted the newborns of high-risk carrier mothers to interrupt perinatal mother-to-child transmission, and quickly expanded it to all newborns, preschool children, and eventually primary school children, halting horizontal transmission.
By tracking the birth cohorts born before the 1984 program and comparing them to the cohorts born after, researchers essentially evaluated a nation-sized community trial.
And the results.
The initial results were exactly what you would expect from a successful vaccine. The carrier rate of HPV among children plummeted from roughly 10% to less than 1%, and infant mortality from acute fulminant hepatitis dropped dramatically.
But the true magnitude of this intervention wasn't just preventing a viral infection. It was preventing the end-stage consequence of the evolutionary spiral.
Years after the program's implementation, nationwide cancer registries revealed a striking, statistically significant decline in the incidence of childhood hepatocellular carcinoma. By deploying a vaccine against a biological environmental agent, they engineered a massive reduction in pediatric cancer.
That's incredible.
The sources explicitly state that this was the first definitive proof in human history of cancer prevention through vaccination. It provided ironclad, reversible proof that chronic HPV infection causes liver cancer.
It is a breathtaking achievement. They intercepted the biological agent before it could even begin the spiral.
Yes.
Both the arsenic water replacement and the HPV vaccination program were essentially uncontrolled community trials, right? The intervention was applied to everyone.
Correct.
But the sources provide a brilliant example of a controlled cluster trial that I want to explore, as it beautifully illustrates how to resolve competing scientific hypotheses in the field. The endemic goiter study.
Ah, yes. Before World War II, endemic goiter, which is the massive pathological enlargement of the thyroid gland, was extraordinarily prevalent in the mountainous inland regions of Taiwan. In some remote villages, up to 70% of the population was afflicted.
70%.
Epidemiologists faced two competing theories. One theory proposed that the environment simply lacked sufficient dietary iodine, which is essential for thyroid hormone synthesis.
And the other theory.
The competing theory suggested the environment was rich in goitrogenic substances, specific compounds found in local dietary staples like cabbage or cassava that actively block the thyroid's ability to absorb iodine.
So to resolve this, they didn't just blanket the entire region with iodine supplements. They designed a rigorous experiment. They selected two distinct mountainous townships that were highly comparable in their socioeconomic status, dietary habits, and baseline goiter prevalence.
Yes.
They then took complete control of the commercial salt supply entering both towns.
In the designated experimental township, the public health teams ensured that only iodized salt was available for purchase and consumption. In the control township, they provided normal, non-iodized salt.
And what happened?
The results were rapid and definitive. After a relatively short period, the incidence and prevalence of endemic goiter in the schoolchildren of the experimental township dropped precipitously, while the rates in the control township remained high.
The controlled structure of the trial isolated the variable perfectly. It definitively proved that environmental iodine deficiency was the primary causal factor overriding the goitrogenic food theory.
Exactly.
This localized experiment provided the evidentiary foundation for a massive, nationwide salt iodization program backed by UNICEF, which virtually eradicated endemic goiters across the country.
These macro-level interventions, water replacement, universal vaccination, salt iodization, are the absolute triumphs of population health.
They really are.
But as we move deeper into the science, we confront a persistent, granular mystery. Even in a township where everyone drank from the same artesian well for 40 years, or in a cohort where everyone was infected with hepatitis B, not every single person developed cancer.
Right. Some live to be 90 without a trace of disease.
Exactly. How do we explain that massive variance in outcome when the environmental exposure is seemingly identical?
To answer that, we have to abandon the macro-level view of townships and reservoirs. We have to zoom in past the organ system, past the tissue, down to the molecular architecture of the host. We have to analyze gene-environment interaction.
Yes, the micro level.
You must understand how the unique genetic code of the individual interacts with the physicochemical agent. The primary tools we use to measure this molecular interaction are biomarkers.
A biomarker is a quantifiable, objective indicator of a biological state. It translates an abstract environmental exposure into a measurable molecular data point. In the realm of environmental health, we stratify these into three critical categories: biomarkers of exposure, biomarkers of effect, and biomarkers of susceptibility.
Let's systematically break those down, returning to the heavy metal we know so well. Arsenic. What constitutes a biomarker of exposure?
A biomarker of exposure confirms that the agent has successfully breached the host's defenses and entered the biological system. We measure the internal dose.
Right.
For acute or recent arsenic exposure, a clinician might measure the concentration of arsenic in the patient's urine, hair, or fingernails. But a much more sophisticated and clinically relevant metric is the biologically effective dose.
How is that different?
This doesn't just tell us the toxin is present, it tells us how much of the toxin is actively circulating in a form capable of damaging cellular machinery. For ingested arsenic, the key biomarker of the biologically effective dose is the relative proportion of a specific metabolite, monomethylarsenic acid, present in the urine.
Okay, so the exposure biomarker proves the agent is inside the gates. What about the biomarker of effect?
These biomarkers measure the actual biological damage or physiological alterations caused by the agent long before the patient develops the systemic clinical disease. They are the subclinical lesions.
Give me an example.
In the context of chronic arsenic exposure, a classic clinical biomarker of long-term effect is the development of skin hyperpigmentation and palmoplantar hyperkeratosis, the pathological thickening of the skin on the palms of the hands and soles of the feet. At a microscopic level, effect biomarkers include chromosomal aberrations, the formation of micronuclei in cells, or elevated levels of oxidative stress markers in the blood. If we use imaging, detecting increased intima media thickness via carotid ultrasound is a biomarker of effect indicating early atherosclerosis, years before a cerebral infarction occurs.
I always explain biomarkers of effect to medical students using the check engine light metaphor.
That's a good one.
The car might still be driving down the highway at 60 miles an hour, seemingly fine, but the internal sensors have detected a critical parameter operating out of range. The light turns on. The biomarker allows the clinician to detect the subclinical lesion in the progression phase of the evolutionary spiral, providing a window to intervene before the engine violently seizes and clinical disease manifests.
Exactly.
But the category that truly unlocks the mystery of individual variants is the biomarker of susceptibility.
Yes, susceptibility biomarkers define the inherent vulnerability of the host's machinery. They can be acquired, for instance. Measuring low serum levels of keratines or other micronutrients indicates a poor nutritional baseline that leaves the host vulnerable to oxidative stress.
Right.
But the most profound susceptibility biomarkers are genetic. With arsenic, the human body attempts to detoxify the heavy metal through a process called methylation, utilizing specific enzymes to process and excrete the toxin.
And genetics affect those enzymes.
Exactly. The genetic polymorphisms, the specific sequence variations in the genes coding for those methylation enzymes, dictate how efficiently an individual can clear the arsenic.
So if a patient inherits the genetic coding for highly efficient methylation enzymes, they can process a moderate arsenic load with minimal cellular damage. But if they possess a genetic polymorphism that renders those enzymes sluggish or ineffective, the arsenic bioaccumulates rapidly, and their risk of cellular toxicity and malignant transformations skyrockets.
Spot on.
It's the molecular manifestation of the ecological wheel, the physicochemical environment pressing against a vulnerable genetic core. And the sources provide a breathtaking example of this by detailing the synergistic interaction between a virus, a chemical toxin, and a specific genetic deletion.
We return to the pathogenesis of hepatitis B-related liver cancer to observe the ultimate cascade of gene-environment synergy.
Okay.
First, we establish the biological baseline using viral biomarkers. We screen the patient's serum for hepatitis B surface antigen, or HBsAg, and E antigen, HBag, to confirm active viral replication. We measure the exact viral load, the quantifiable copies of HBV DNA in the blood. The higher the viral load, the more aggressive the baseline inflammation.
But the evolutionary spiral is multifactorial. The host is also navigating their chemical environment, specifically their dietary environment.
Yes.
The textbook details the severe impact of aflatoxins. Aflatoxins are fiercely potent chemical hepatotoxins produced by specific fungi like Aspergillus flavus, which routinely contaminate agricultural crops like peanuts and corn in warm, humid climates.
To measure the biologically effective dose of this chemical agent, researchers look for a very specific molecular footprint in the blood. AFB1 albumin adducts.
Adducts.
Yes, an adduct occurs when a reactive molecule of the toxin physically binds to a protein or DNA strand, altering its structure. Massive observational studies demonstrated a clear, dangerous synergy.
What kind of synergy?
Individuals who are chronic HBV carriers and who simultaneously have high dietary exposure to aflatoxins have a significantly elevated risk of developing hepatocellular carcinoma compared to carriers with low aflatoxin exposure. The viral biological agent and the chemical agent act in concert to assault the hepatocytes.
So the virus establishes the chronic inflammation, forcing the liver cells into a state of hyperproliferation to repair the damage. Meanwhile, the aflatoxin directly attacks the DNA of those rapidly dividing cells, inducing massive mutations.
Exactly.
But where does the biomarker of susceptibility fit into this crossfire?
This is where the intricacies of xenobiotic metabolism, the way the body detoxifies foreign chemicals, become critical. The human body has an arsenal of enzymes designed to neutralize toxins like aflatoxin. A critical family of these defense enzymes is the glutathione S-transferases, specifically encoded by the GSTM1 and GSTT1 genes.
Okay.
These enzymes work by conjugating the toxic molecule with glutathione, rendering it water-soluble so it can be safely excreted in the urine. However, a significant percentage of the population inherits a genetic anomaly known as a null genotype for these genes.
As clinicians, when we see the term null genotype, we know exactly what that implies structurally. It means the specific gene is entirely deleted or completely non-functional. The host has zero capability to produce that specific detoxifying enzyme. The metabolic pathway is completely dark.
Exactly. They lack the molecular defense shield against that specific class of toxins. And when epidemiologists layered this genetic susceptibility biomarker over the viral and chemical exposure data, the results were explosive. If you observe chronic HBV carriers who possess the normal wild-type GST genes, their risk of liver cancer increases moderately when they are exposed to heavy dietary aflatoxins. Their enzymes are actively fighting a rearguard action against the chemical assault.
Right, they have defenses.
But if you observe chronic HBV carriers who possess the null genotype for GSTM1 or GSTT1, and they're exposed to the same heavy dose of aflatoxins, their risk of hepatocellular carcinoma undergoes a massive synergistic amplification. The risk doesn't just add, it multiplies exponentially.
It is the perfect catastrophic storm on the evolutionary spiral. The biological virus degrades the tissue, the chemical toxin fundamentally mutates the DNA, and the host's genetic lack of an enzymatic defense means the chemical assault proceeds entirely unopposed.
It's an incredible piece of epidemiological detective work.
But it presents a daunting clinical challenge. When I am sitting in an exam room with a patient, I cannot simply hand them a scatter plot of 30 different viral, chemical, and genetic variables and expect them to understand their risk.
Yeah.
We have to synthesize these molecular chaos into actionable clinical data.
And that synthesis is achieved through the development of multivariable risk calculators. A risk calculator utilizes complex regression models to integrate multiple independent predictive biomarkers into a single cohesive statistical tool.
Okay.
It calculates an individual's absolute cumulative risk of developing a specific disease over a defined period of time. It resolves the clinical paralysis of dealing with several marginal risk factors that individually might not trigger aggressive intervention but collectively represent a severe impending threat.
The sources detail a remarkable, internationally validated tool derived from the REVEAL HBV cohort study, a risk nomogram for predicting hepatocellular carcinoma in chronic hepatitis B patients. How does a clinician practically deploy this nomogram?
Well, the nomogram visually translates complex statistical weightings into simple integer points. The clinician evaluates the patient against a series of specific predictive variables. The model assigns points based on age, sex, family history of hepatocellular carcinoma, specific liver enzyme levels, namely serum alanine aminotransferase, or ALT, the patient's HbA serostatus, the quantifiable viral load, and the specific genetic genotype of the hepatitis B virus they harbor.
Let's run the math on a hypothetical clinical scenario provided in the text. Let's say I am evaluating a 64-year-old male patient who is a chronic HBV carrier.
We systematically score his biomarkers. He falls into the 60 to 64 age bracket, which the model heavily weights, assigning him 6 points. His blood work shows an elevated serum ALT level of over 45 IUL, which indicates ongoing liver inflammation and adds 2 points. He discloses a family history of liver cancer, adding another 2 points. Lab results confirm he is HB-big seropositive, and his viral load is exceedingly high, over 1 million copies combined with a genotype C viral infection. That specific virological profile adds a massive 7 points to his score.
The statistical weighting is fascinating. The baseline inflammation of an elevated ALT only adds 2 points, but the sheer volume of the replicating virus adds 7 points. It tells the clinician exactly which biomarker is driving the pathology.
Right.
So we sum his scores. 6 points for age, plus 2 for ALT, plus 2 for family history, plus 7 for the viral load profile. His total risk score is 19. As his physician, how do I interpret a score of 19?
The clinician plots that integer sum of 19 onto the nomogram's survival axis to derive the patient's cumulative risk. For a score of 19, his 5-year cumulative risk of developing hepatocellular carcinoma is roughly 40%. His 10-year risk doubles to 80%, and his 15-year cumulative risk is an overwhelming 90%.
Wow. It is a staggering clinical tool. It takes abstract population-level epidemiology and distills it into a highly personalized, terrifyingly precise weather forecast for the individual sitting on the exam table.
Very accurate.
If I tell a patient they have an 80% chance of developing a lethal malignancy in the next decade, that entirely dictates the aggressiveness of our management. It justifies immediate initiation of potent antiviral therapies. It mandates intensive abdominal ultrasound screening every 3 to 6 months, and it ensures total patient compliance. It perfectly bridges the gap between the macro-ecological study and the micromolecular intervention.
It represents the pinnacle of targeted, personalized medicine. However, as we synthesize the overarching lessons of this deep dive, we must zoom our focus out from the individual clinical encounter. We must return to the macro level, to the global ecosystem.
Why is that?
Because while risk calculators manage the disease once the spiral has begun, the ultimate goal of public health is to ensure the spiral never initiates. The promotion of health through environmental intervention is a massive systemic imperative.
We cannot medicate our way out of a toxic environment.
We really can't.
And crucially, the physical, chemical, and biological environments do not respect sovereign borders. Globalization ensures that an environmental hazard generated in one hemisphere is a shared burden for the entire planet.
The examples are ubiquitous. Industrial air pollution, laden with particulate matter and polycyclic aromatic hydrocarbons, is generated in one nation, but rides the prevailing winds to induce severe respiratory and cardiovascular morbidities in neighboring countries.
Right.
Agricultural runoff, heavily saturated with nitrogen and phosphorus, pollutes transnational river systems and shared oceans, destroying marine food webs. The global emission of greenhouse gases drives anthropogenic climate change, which fundamentally alters the temperature and humidity baselines of entire continents, aggressively expanding the geographic habitats for vector-borne diseases like dengue and Zika. Environmental stewardship is fundamentally a borderless necessity.
And what is vital for medical students and public health trainees to internalize is that upstream environmental interventions are not just ethically imperative. They are incredibly systemically cost-effective. Engineering the hazard out of the environment yields massive secondary and tertiary dividends that extend far beyond simply recording fewer sick people in a hospital registry.
The ripple effects of robust environmental health policies are profound and structural. When a public health initiative secures a pristine, accessible water supply for a rural township, they do far more than just interrupt the transmission of cholera or prevent the vascular necrosis of Blackfoot disease.
They strike a blow against extreme poverty.
Exactly. They promote gender equality and universal education because children, disproportionately young girls, are no longer forced to spend 4 hours a day walking miles to fetch contaminated water instead of attending school.
Safe environments drastically reduce child mortality and severely lower maternal morbidity. Creating a stable, non-toxic baseline of environmental sustainability creates a resilient population, making the control of other concurrent epidemics, such as HIV, malaria, and tuberculosis, fundamentally easier and cheaper to manage.
The overarching, inescapable conclusion of the source material is that human biology is entirely porous to the world around it. The health of a human population cannot be sequestered or separated from the health of the ecosystem it inhabits. We are, at a molecular level, the air we breathe, the water we drink, and the soil that grows our food.
Precisely. And that intimate, inseparable biological connection is exactly why the study of environmental health remains one of the most dynamic, complex, and absolutely critical disciplines within the broader architecture of global public health. Which brings us to the close of our conceptual deep dive. But before we move into the structured learning reinforcement to solidify these frameworks, I want to leave you with a final provocative thought regarding the future of the evolutionary spiral.
Oh, this is an interesting angle.
Historically, as we've discussed at length today, public health has aggressively focused on the physical, chemical, and biological environments. We measure the heavy metals in the artesian well, we measure the particulate smog in the air, we track the viral load of the pathogen. But moving forward into the mid-21st century, human populations are spending massive, unprecedented, and uninterrupted portions of their lives immersed in entirely synthetic digital environments.
Digital environments, yeah.
As public health practitioners looking to the future, how will we define the miasma of the internet? What will be the quantifiable biomarkers of effect for continuous exposure to algorithmic ecosystems? Will we measure the downregulation of dopamine receptors? Will we track the alteration of pediatric neuroplasticity as a measurable subclinical lesion? How will the invisible constant exposure to digital environments synergize with our genetics to shape the chronic, non-communicable diseases of the next 50 years? It is the absolute bleeding edge of environmental health, and it is something crucial for every clinician to begin considering.
A profoundly relevant and deeply complex question for the next generation of epidemiologists. Now, to ensure the dense foundational concepts from today's rigorous discussion have firmly anchored in your memory, we will conclude with our rapid single best answer prompts. I will read the clinical question and the four options. I will pause briefly to allow you to actively retrieve and lock in your answer, and then I will provide the correct response along with the specific chapter section you should revisit for further review.
Take it away.
Question 1. Which causal model is most appropriate for structurally describing the multi-stage temporal pathogenesis of a chronic disease such as hepatocellular carcinoma that involves a complex, decades-long interaction between a viral agent, chemical toxins, and host genetic susceptibility?
A. Epidemiological triangle. B. Ecological wheel. C. Evolutionary spiral. D. Cox postulates. Pause.
C. Evolutionary spiral. If you need to review the dimensions of time and multi-stage pathogenesis, please revisit the section on causes of human diseases.
An epidemiological investigator observes a highly significant, strong, positive correlation between a rural township's average artesian well water usage and its aggregate bladder cancer mortality rate. If a clinician assumes that this aggregate statistical relationship perfectly and uniformly applies to the individual risk of a specific patient in that township, without adjusting for individual-level confounding variables like smoking history, they are committing which methodological error?
A. Selection bias. B. Ecological fallacy. C. Recall bias. D. Confounding. Pause.
B. Ecological fallacy. To review the critical limitations of aggregate data analysis, revisit the section on ecological studies.
In the clinical assessment of a patient chronically exposed to environmental arsenic, determining the biologically effective dose is paramount for evaluating the active internal hazard. Which of the following is the most appropriate and specific biomarker for measuring the biologically effective dose of ingested arsenic?
A. Total arsenic concentration in hair follicles. B. The clinical presentation of palmoplantar hyperkeratosis. C. The relative proportion of monomethylarsenic acid in the urine. D. The acute concentration of arsenic in whole blood. Pause.
C. Monomethylarsenic acid in urine. This specific metabolic biomarker is thoroughly covered in the gene-environment interaction section.
The dramatic, statistically significant decline in the incidence of childhood hepatocellular carcinoma observed in Taiwan in the years following 1984 was primarily achieved through the implementation of which specific environmental health intervention?
A. The infrastructural replacement of artesian well water with surface water. B. The deployment of a universal hepatitis B vaccination program for newborns. C. A mandatory nationwide salt iodization program. D. Strict regulatory limits on agricultural dietary aflatoxin contamination. Pause.
B. Universal hepatitis B vaccination program for newborns. You can review the methodology and massive public health impact of this achievement in the intervention trials section.
A patient who is a chronic hepatitis B carrier resides in an area with high dietary exposure to hepatotoxic aflatoxins. Their absolute risk of developing hepatocellular carcinoma undergoes a massive synergistic amplification if they possess a genetic null genotype that eliminates their enzymatic capability to detoxify the chemical agent. This null genotype most likely affects which of the following gene families?
A. Androgen receptor genes. B. Glutathione transferase genes, GSTM1, GSTM1, GSTT1. C. Cytochrome P450-1A1 genes. D. DNA mismatch repair enzymes. Pause.
B. Glutathione transferase genes, GSTM1, GSTT1. This critical example of molecular gene-environment synergy is extensively detailed in the gene-environment interaction section.
Excellent work. Thank you all for dedicating your time to mastering the rigorous frameworks of environmental epidemiology and for joining us on this exploration of the invisible physical scaffolding that shapes human health. Here are the details of this episode.
Environmental health. Byline. Air, water, soil, and systems, understanding how the environments we build and inhabit shape human health.